Okay, good morning. My name is James Stanger, and this is my first video on euploid embryos, which I've updated to correct a few typos and add a bit more content. So, this is a new series of videos concentrating only on studies using euploid embryos. They explore basic embryology, embryo behaviours, and implantation. It excludes the impact of aneuploid embryos, especially on pregnancy rates. I will discuss several recent publications that will explore what conditions or observations are related to euploidy and what may not be, and what the implications for it. Just remember that euploidy is currently determined by BGTA, and that may induce some flaws. Non-invasive PGTA may or may not provide a different outcome. So, there are two different viewpoints on euploid embryos, one if you're a staff member and one if you're an IVF client. From a staff point of view, euploid embryos are determined after pre-implantation genetic testing for aneuploidy. They represent viable embryos for transfer and storage. They may either be euploid or minimally mosaic embryos. They are identified by the biopsy of trophectoderm and not the inner cell mass. They may not truly represent the future health of a baby, but they have been shown to have 50% lower incidence of miscarriage and a shorter time to pregnancy. Since after transfer they are most likely to result in a live birth, many studies are now concentrating only on euploid embryos. So what can they tell us about implantation in IVF? Is this a potential new standard? If, on the other hand, you're a client, a patient coming through using the services to fall pregnant, the critical information you need to know about euploid embryos is one, about 40% of all embryos have the correct number of chromosomes, and that's called a euploid embryo. They can only be determined after genetic testing or biopsy of the embryo on day 5 and then testing, then vitrification, freezing. Euploid embryos have a lower chance of miscarriage and a shorter time to pregnancy. But however, some embryos labelled as not euploid, that is called aneuploidy, may be overestimating the likelihood of the inner cell mass which gives rise to the baby being abnormal. So it may be that some embryos labelled as aneuploidy and not transferred may in fact result in the birth of a live baby, but with a reduced outcome, likelihood of success. Some embryos are a mixture of euploid and aneuploid cells, these are called mosaic, and the degree of mixture can reflect the health of the future child and the likelihood of pregnancy. So I'm going to review 4 papers looking at pronuclear dynamics, mitotic errors in the trophectoderm, the frequency of euploidy amongst different cohorts of blastocysts, and the using the inner cell mass as a tool to give you the highest pregnancy rate. I've included another one here looking at the impact of morphology-based selection for male skewed sex proportion, just as a counter to this publication. Okay, let's start. The first paper I want to talk about is a non-access, open access paper published in Human Reproduction by an Italian group. And these people were looking using time-lapse imagery, saying the non-pronuclear fertilization pattern is assessed by a single static observation that most scientists do between 16 and 18 hours post-insemination interval. Are they compatible with embryo development competence? So this is a broader publication of which, but they're looking at the impacts of euploidy. So the answer is a few zygotes, that's underlined, who show pronuclear solely outside the static observation time can develop into blastocysts and euploid blastocysts, and there are rare cases of cleavage without pronuclear formation, that these nearly always result in early development arrest and degeneration. So I'm only going to focus on one little bit, which is the relationship between when the pronuclei appear and disappear. So once again, this is a non-open access paper, and I'm going to display 2 tables from that. I hope it's okay. First is that when they look at an odds ratio analysis, maternal age is clearly associated with 2 PNs forming blastocysts. But what's interesting, and our scientists should take note here, is that if you use sequential media, the continuous media comparison produces more blastocysts, significantly more blastocysts. So for 30, 40 years, we've been using sequential media to grow our embryos from day 1 to day 5. But since we've been using time-lapse imagery, the time-take incubator, and they need to keep the embryos in the same media, this media has been proving to be superior in producing more blastocysts. However, the case in point, the pronuclei appear somewhere 16 to 18 hours, 6 to 8 hours after insemination. So if you compare the embryos where the pronuclei appear early, if they appear later, 16 to 18 hours or after 8 hours, the odds ratio for forming blastocysts significantly falls away. So any embryo that forms pronuclei after 18 hours has got a very, very low probability of forming a blastocyst. Conversely, if you look at when the pronuclei fade, if the embryos fade between 18 and 20 hours post-insemination, the odds ratio is 1.6 really high that you are going to get a blastocyst. But where they fade significantly sooner, before 18 hours, the odds ratio for forming blastocyst falls away very quickly. So that is slow pronuclear development is associated with poor blastocyst development. And fast pronuclear completion is associated with better blastocyst development. That's the take-home message. But these are blastocysts, not euploid blastocysts. I just need to provide some illustration for the non-IVF clients. This is an oocyte on day 1. It's called a zygote. And proof that's been fertilized are these two circles. They're called pronuclei. And one comes from the female chromosomes and one comes from the sperm. And there is a development where they suddenly appear, they grow, they abut very quickly, and then they disappear. So we're looking at the time when these pronuclei first appear and when they disappear. So take some pictures from a time-lapse video. You can see here there's a single pronucleus very early on and no sign of pronuclei. And by a short period of time, later 6 to 8 hours, you have your second polar body being extruded, and to see two small pronuclei just starting to appear in the cytoplasm. This is appearance, time of appearance. They get bigger, and then they start to get close together, then they abut. Of course, the time lapse, you can't orientate the profile which to get the best view of it. So sometimes the picture of pronuclear development is poor in time. That's one of the disadvantages. That's what you're looking at from this point of view. Then all of a sudden, around about 18 hours, they just disappear, boom, very quickly. They abut very close, they pull very tightly together, and then the membrane disappears very quickly, and the chromosomes, the egg and sperm, first mix for the first time. This is the true new embryo, and very quickly after that, the embryo divides in its first mitotic division to the 2-cell stage. But if you go back and look at this data, not only for blastocysts, but for euploid embryo, euploidy, the relationship with age still exists, but when the embryos appear is less clear. So comparing embryos where the pronuclei appear before 16 hours to when there's delay, you can see that the difference is not significant. Even though there appears to be a better, the difference is not significant. However, if you look at the time when pronuclei disappeared, 20 hours, compare that to 18 to 20, there is a highly significant increase in the likelihood of that blastocyst being euploidy, and less so if the pronuclei appear before that. So after adjusting for maternal age, only the time of fading 6 to 18 hours shows a significantly higher euploidy rate than if the pronuclei fade after 20 hours. So the comment here is the rate at which pronuclei develop and disappear is loosely related to blastocyst development and to a less extent of the euploidy rates. Prior to their appearance, the time spent unraveling of the chromosomes and for the male, the replacement of the protamines with histones takes a long time. Pronuclear, it's really amazing. It gets done within, 10 hours or so. The pronuclear stage is also a period where the embryo is checking the DNA and starting to activate the embryonic genome. Damaged sperm DNA and slow oocyte activation by phospholipase C are two recently identified factors that may contribute to delays. There seems to be a strict time for pronuclear appearance but a slightly larger range for pronuclear disappearance and euploidy, except that statistically, the best time is in this sweet spot here, 18 to 20 hours. So if you're going to do IVF or ICSI without time-lapse imaging, you really do need to be looking at the embryos no earlier than 16 and no later than 20, but 17 to 18 hours still is the best time to check to see if prenuclei, what they look like, and you may need to come back 2 hours later to look at time, which ones, when they're disappearing. And just to finish up, since almost all aneuploidy is due to oocyte meiosis errors there, how does the rate for prenuclear development influence the euploidy rate? One way of looking at it is, this is my comment, is that euploidy in the current IVF vocabulary means true euploidy plus low-level mosaic embryos. Therefore, one explanation may be that perturbations in the fertilization process may lead to increased mitotic errors in the embryo as it starts to cleave, and especially inter-affected after compaction. Perturbations may include oocyte maturity, sperm DNA damage, or delays in sperm incorporation into the oocyte after ICSI. And I'd just like to make another paper here, which is published just recently in Journal of Assisted Reproductive Genetics. It's a good journal, that one. Looking at the rate at which blastocysts develop compared to the morphokinetics, basically saying that paternal pronuclei appear earlier in day 4 blastocysts than in day 5, whereas their appearance when it was delayed also resulted in delayed blastocyst formation on day 6. So when the paternal, that is, usually you can tell that's the larger one of the two, when the paternal DNA appears early, you get earlier developed blastocyst formation, and when the pronuclei appears later, you get delayed blastulation. In paternal and maternal pronuclei, the time interval from pronuclear appearance to breakdown was shorter in day 4 blastocysts, but prolonged in day 6 and 7 and 8. And the question is, does that really make a difference in terms of live birth? And for day 6, probably not. For day 7, definitely. Okay, I'd like to go into the second paper, which is published in Fertility and Sterility Reports, Open Access. And this also is a good paper. They're looking at euploidy rates within blastocysts obtained from different size cohorts and age groups approximate a binomial distribution. So the objective of this paper was to investigate whether the distribution of euploid blastocysts in terms of cohort of embryos that can be biopsied can be predicted. And the conclusion was the probability of distribution structure of euploids within discrete age cohort sizes is binomial for all age group and for nearly all cohort sizes. The probability, now this where you have to pay attention. I think it's a poorly written conclusion. Probability of distribution ranges identified represent a more accurate frame of reference that describes and predicts the number of euploid blastocysts most likely to be present within an individual cohort of blastocysts before biopsy, with estimates from simple age group over averages. And this is what the data is all about. So it's actually quite interesting when you focus on it. I've summarized another table they had, just looking at the number of euploid blastocysts per blastocyst that were biopsied by age. And overall, they were reporting a 61% euploid blastocysts per blastocyst. So they're saying almost 2/3 of all embryos biopsied will be euploid. But if they're under 30, that's almost 3/4. And it falls a little bit within 30 to 34 and 35 to 37. And even 38 to 40 is still 4 chances in 10 of getting a euploid from blastocyst biopsied. But then you hit the wall, and by 41, only 1/4 of all blastocyst biopsied will be euploid. And if you're over 42, that drops to less than 1 in 5. Now, what's interesting, if you go into the different age cohorts, I think these are based on the USA SART group. This is the number of blastocysts available for biopsy. So in this case, there's 1 available, 2, 3, 4, 5. In this case, there were 10 or more blastocysts available for biopsy. Fancy having 10 blastocysts to biopsy. I feel sorry for the poor embryologist. I also wonder just how many eggs and embryos they collected. But this is for women under 30. And increasingly, these are a rarer group of patients coming through. But you can see the likelihood of getting a euploid embryo from a biopsy blastocyst was fairly linear. So in this case, if you've collected enough eggs and embryos to give you 10 blastocysts on day 5 for biopsy, although it could actually also include day 6, I suspect, they may realistically expect at least 8 of those 10 or more embryos biopsied were to be euploid. And I would love to see the cumulative pregnancy rates for this group of under 30-year-olds being looked at in more detail and seeing how close they can get to 100% pregnancy rate. Because you'd like to think that if you had eight euploid embryos from a case and you put one embryo back in 8 different transfer cycles, you'd like to think that your pregnancy rate was at least 100%. And they came back for subsequent pregnancies. It'd be really interesting looking at that data. I don't think anyone's does. For 30 to 34-year-olds, you can see it's also fairly linear, but not quite as harmonious as under 30. Once again, it slopes a little bit more, a little bit less. So that for 35 to 37-year-olds, if you got a high AMH and managed to get 10 blasts for biopsy, your chance of getting euploid is roughly about half. It falls to even less than that, just over 1/3 if you're in the 38 to 40. And if you only have 2 or 3 or 4 embryos, 38 to 40, your likelihood of getting a euploid embryo for transfer starts to become of concern. But once you go through this doorway, this paradigm shift to 41-year-olds, look at this. All these embryos, they're being biopsied. And it's only this case of 7 or 8 or 9 do you start to get more than one embryo being euploid. And if you're 33 or 43 or more, you may as well just not bother. So the probability that a blastocyst is euploid decreases with maternal age and increases with the cohort size of blastocysts can be biopsied. And I just want to make a note here that this is euploid, but when they say 61%, I suspect they will also be including mosaics of somewhere between 10 to 40%, depends on the clinic, how they interpret, etc. So it may not be a true euploid, it might be euploid plus low-grade mosaics. So they concluded the number of euploid blastocysts is largely linear to blastocyst cohort in size. This is largely age-related, with older women having fewer euploid embryos per blastocyst than younger women. So age impacts both on the number of oocytes and embryos available and fewer euploid embryos per blastocyst biopsied. Older women opting for PGTA are increasingly less likely to have a viable embryo for transfer. They need to recognise that. However, younger women under 30, maybe under 35, may reasonably expect 1 to 2 in every blastocyst. There's another typo. Another 3 blastocyst biopsy will be sufficient for transfer. Okay, the third paper is looking at the incidence of mitotic errors in pre-implantation embryos. So this is published in Nature Biotechnology, late October. A new imaging technique allowed scientists at the University of Cambridge to observe that some genetic errors appear late in embryo development than previously thought, and were present only in cells destined to become only in cells that were trophectoderm that were destined to become part of the placenta. This means that some embryos flagged as abnormal may still be viable, since the adrenal cell mass may well be euploid, and therefore could influence how clinics select embryos in the future and how they can transfer or discard. They discovered that some chromosomal abnormalities arise after the cells have already differentiated into trophectoderm, that is once become placenta, from cells that have already going to form the inner cell mass, which will form the fetus. However, the research shows that embryos can have changes in these cells that are not present in other parts of the embryo that would become the fetus, thereby giving a potential misleading picture of the embryo's overall viability. As a result, the study suggests that some embryos may be discarded unnecessarily, potentially affecting patient outcomes and costs. Our findings challenge the current practice of trophectoderm biopsy for pre-implantation genetic testing for aneuploidy, a widely used clinical method to detect aneuploidies in human embryos. It's generally assumed that the aneuploidies detected originate from early meiotic or the first lot of mitotic divisions. However, the authors provide evidence using advanced chromosomal tracking and live embryo imaging and segmentation technologies to show that de novo mitotic errors can still arise late in human pre-implantation development, thereby clouding the issue of when you do biopsy of the trophectoderm. Our data supports the conclusion that these errors may be confined to the trophectoderm, leaving the inner cells unaffected. They also observed periodic cycles of blastocyst growth and collapse, which were accompanied by fluctuations in nuclear volume. And I think they're talking about this because this may well affect the number of cells that they can see. These dynamic changes may be driven by mechanical forces, such as those arising from blastocyst expansion, hatching, and collapse. The current labelling method predominantly marks trophectoderm cells, and as a result, it's not yet clear whether similar changes occur in cells in the inner cell mass at this stage. Now, this was just a comment, but it's an important one because they're looking at trophectoderm and saying a lot of cells can become aneuploidic, have a mitotic error as they're dividing. Sorry, I just got to put my volume back up. But they're not really looking at the inner cell mass. So we actually don't know what happens in the inner cell mass, whether there are mitotic errors or not, and what happens to those cells. And that's a more interesting question. Such embryos could be called mosaic, and as indicated before, many embryos defined as euploid may contain up to 40% mosaicism. So this article suggests that some mosaicism is due to late-stage mitotic errors. So some of these errors, some of these embryos that they're looking, they can see mitotic errors occurring in the trophectoderm may well have been quite normal when they first started to cleave. So the inner cell mass may well be quite normal. The second last paper I'd like to look at rather mischievously is 2021. They're basically saying that morphology-based selection from available euploid blastocysts induces male-skewed sex proportion in the offspring. So this is about rates of division, the morphology of the trophectoderm, and morphology in the cell mass. And I'll give you two papers giving diametric opposite outcomes. And this paper says that the sex proportion was impacted by the day of biopsy, day 5, day 6, day 7, and trophectoderm morphology, but not by the ICM morphology in PGDA cycles. Therefore, biopsy on day 5 of embryos with a really good trophectoderm shifted the sex proportion towards males. This was noted on open access files, so there's a limit to what I can show you. The results, though, it's kind of interesting. They talk about this sex proportion was impacted by the day of biopsy and trophectoderm morphology, but not ICM morphology. Therefore, biopsy on day 5 with A-grade trophectoderm shifted the sex proportion towards males. Interesting, though, they noted that our morphology-based embryo selection for single embryo transfers of euploid blastocysts had favoured the choice of XY embryos, generating a 54% to 56%, 54% skewing of transfer and 50% skewing at live birth. It's interesting. They talk about this, but it's only changing the sex ratio by about 3%. It's not something that you base your future gender baby on. It's just noting that there is a slight bias that male embryos do grow a little bit faster, and they put a lot more emphasis on trophectoderm. And in a funny sort of way, this is actually making the trophectoderm important, is confirming that the fertilization was a sexual fertilization and not an internal one. So the models indicate a weaker association between blastocyst morphology parameters and sex proportion in non-PGDA cycles. So even when you're not doing PGDA, so if you're just picking out embryos based on their morphology, trophectoderm, and how well they're dividing, you do get a slight skewing of males, but it is not significant. On the contrary, this paper published in October this year argued that the inner cell mass and early blastulation are associated with pregnancy outcomes in euploid embryos. So, in this paper, the embryo sex was known in 90% of transferred embryos, with the result of 51% were females and 49% were males. That is, even when you knew the gender of the embryo from your PGA testing, in reality, the embryos you transferred were basically one-to-one, a slight skewing of females. In 161 cases, or 21% of their cycles, where the embryo sex was known and was used to select the embryo for transfer, even in those situations, you're only skewing the sex ratio to 52%. And they published this paper, which is kind of interesting. This is looking at the difference between morphological measures and live birth by embryo sex. And for male embryos, the day of vitrification, as they were going fast or slow, was significantly related. Odds ratio 0.7. So the embryos that were vitrified on day 6 and 7 had a lower pregnancy rate than those ones that were vitrified on day 5. And the quality of the inner cell mass or trifectoderm was unrelated. For male embryos, it was just how fast they got to a blastocyst stage. But for female embryos, the rate of division wasn't as important, but the quality of the inner cell mass was highly significant in terms of the live birth rate. And where the inner cell mass was poor, quality 2 or 3, the odds ratio was only 0.6. So compared to class quality 1, a very poor inner cell mass was associated with a significantly lower pregnancy rate, implantation. And the last table they presented looking at the live birth rate by embryo sex, quality inner cell mass, and the day of vitrification. Basically, if you ICM1s are the best ones, they have a well-formed, tightly packed inner cell mass. If you're picking embryos out with the best inner cell mass on day 5, 60% will be female, and 56 will be male. If you choose on embryos with a poorer inner cell mass, the rate's actually fairly similar, about 40%. Still, 40% is pretty good, but it's not 60%. If you let the embryos divide one more day, that is, they're dividing a bit slower, the inner cell mass grade, top grade, gives 54% for females and 41% for males. So there is a difference. If you're actually looking, you're letting an embryo grow out to day 6 because it's growing a bit slow, and you pick an embryo out by its inner cell mass rather than just trifectoderm. No, if you pick an embryo out by quality of the inner cell mass, it's more likely to be female and more likely to give you rise to a pregnancy. And embryos on day 6 with poor quality inner cell mass produce only a pregnancy rate, so sort of half of what the top bit is. So they found that both day of vitrification, the quality in the cell mass and vitrification were significantly associated with the odds of live birth in euploid embryos. In contrast, the quality of the trophoblast of vitrification, the sex of the embryo, and whether an embryo was selected for transfer or not was not really associated. It's all really got to do with the inner cell mass. I think we've had far too much focus on the trifectoderm since we're doing the biopsy, but it's inner cell mass which is telling you quality. So it's basically saying that an embryo that has is divided sufficiently fast, there's enough cell mass available to allocate a significant quality to the inner cell mass. They're the embryos which are most likely to give you a live birth. And if the embryo is starting to blastocyte before it's had enough cell division, that's going slow, and the inner cell mass has been poorly populated, then the chance of getting pregnancy is significantly less. Okay. So in summary, there seems to be a strict time for pronuclear appearance and a larger range for disappearance in euploidy. The number of euploid blasts is still largely linear to the blastocyst cohort size, but they're largely age-related, with older women having fewer euploid embryos than younger women. The de novo mitotic errors can arise late in human prenatal development. The conclusion is that these errors may be confined to the trifectoderm, leaving the inner cell mass unaffected. Sorry, I was just being interrupted. The sex proportion was impacted by the day of biopsy and trifectoderm morphology, but not inner cell mass in PGA day cycles. However, the biopsy on day 5 of embryos with a good trifectoderm shifted its proportion towards males, but if you're after implantation, the euploid embryos with better ICM quality and those reaching the blastocyst stage earlier were associated with the higher odds of live birth, which is what it's all about, folks, suggesting that prioritising embryos for transfer by these parameters may provide patients with the best chance of live birth, and I suspect a slightly skewer towards the number of females. So just to finish up, thank you for persevering. My name's James Danger. I'm an embryologist with more than 50 years experience. I'm not a medical person, and this information is not medical advice, but it may help you in knowing what questions to ask about your treatment with your doctor, and maybe to help in understanding the advice that you receive. If you like what I'm doing, you click subscribe, press like, and make a comment. And you always go fund me, go to my fund me and support me. And one quick word at the end, just saying my other activities, I run a online EQA program for scientists in andrology and embryology. And that if you're a client coming through a clinic using my FertAid site, it may give you some confidence that the skills of the laboratory staff processing your gametes are under constant review and assessment. And the schemes provide 12 iterations a year. And these andrology and embryology schemes, you can enroll as a trainee or a professional. You run small clinic or large multi-lab clinics and use for training or competency or accreditation. Okay. And that, my lot, is what we say is your lot for the week. I'd like to thank you for perseverance. I know IVF is a personal journey that involves many action and many techniques and staff. It helps to understand all aspects of that journey. I hope this information may be of value. If you like what I'm doing, tell other people about it and press subscribe. Okay, I'll say no more.